Hello everyone, this is Shankar Kartekhai from Nature's Laboratory, UK, and I hope till now you must have enjoyed and learned lot about the propolis. Uh, so today I will just go straight to uh, my presentation and focus on the actual study performed and skip the introduction part. So today I'm presenting on Propolis activity factor, which we derived by using multivariate data analysis on the Propolis from various regions. So as you see in the picture, we have derived the we have classified the propolis into three different classes: tropical zone, central part, the subtropical zone, and the zone which is showing in green as a temperate zone. So the samples from polar and subpolar we merged with the temperate, as the number of cases for those regions were very minimum and less than 20, so less than 10. So that was not fitting into the requirement of the statistical tool as in the SPSS software. So we had to merge it with the temperate zone. Sorry. So these are the countries sorry from where the propolis samples were analyzed by different researchers and then published in the form of article and other reports. So we used them. So you can see these are countries where the samples were analyzed. So we performed a SPSS analysis by using 23rd version, and we decided to take anti-microbial activity and antioxidant activity as a dependent variable. The independent variable were the chemicals which are generally found in the propolis, commonly distributed across all climatic zones. As these are the chemical structure of the markers which we selected as an independent variable, and the cinnamates which we clubbed together as one parameter because these chemicals were found. So they are structurally related and more or less biologically also related. So we merge them as a one parameter, independent parameter as a cinnamate in the study. So while selecting the literature and rejecting the literature, the studies which we wanted to incorporate in our model, so we had set some parameters. So something like if the biological activity, antimicrobial activity of a particular sample where the MIC is higher than 500 microgram per ml, we rejected that. We selected the studies below reporting activity below 500 microgram per ml. Similarly, IC50 at for antioxidant activity, which was 200 microgram per ml. We selected those study. We were performed by using HPLC as an analytical tool, and rejected studies performed where the analysis or the quantification was done by using any other analytical technique. So likewise, for antimicrobial studies, we selected those reports where the Staphylococcus aureus was used as a strain of microbe for. And then similarly for antioxidant activity, we selected those studies where DPPS was used as a enzyme for the study. And then the rest other studies where these criteria were not met, were not used for the study in the model, as to in order to minimize the errors. So coming towards the result part, what after running the statistical analysis, after feeding in all the data in SPSS software, and we perform the analysis, run the test, and what we found was the phenol content and flavonoid content was higher in subtropical and temperate region, and in contrast, the activity of a tropical region, antimicrobial activity was higher as compared to the total phenol content and flavonoid content compared to other zones. So that signifies the importance, the fact that we all know the temperate tropical is it's like hot and humid region where the microbial load is higher. So plant are we guess that plant will synthesize more chemicals which are having antimicrobial potential and ultimately end up with the in the propolis by bees. So this fact was this was our hypothesis, our understanding in the beginning, and then that was confirmed by the analysis, performing the analysis using statistical tools. So in this graph, what you can see is the the concentration of individual chemical present in the propolis sample. So this is a median value, and what you can see here, the amount of chrysium, pinosembrin, and pinavaskin is relatively higher in subtropical and temperate region, and the concentration of galangin and cinnamic acid derivatives and cinnamates in the tropical region. So these cinnamates are known for their antimicrobial activities. Yeah, so moving towards next, this is a screenshot of Spaces software. Most of you, I hope, must have seen or maybe used some other statistical tool in your various studies. So in this case, what happened? We used 5 models. So for imputation, because many cases, valid cases were missing, and that leads to problem in the output. And a lot of errors can happen. So the statistical software can impute the values by using cross-referencing, by using the cross-referencing the samples, the values you have fed in earlier. So these values we have taken in our studies are the median of 5 models. And for antimicrobial activity, linear regression analysis was performed, and these are the mean values we found. I will not describe these tables in detail, and I will directly go to the results and explain the. So here you see better the R value, closer the value towards 1, better the results, and the model P value suggested that the model is significant. And here also you can see the significant values as the values are less than 0.05. And this is a coefficient equation which we derived by running the coefficient analysis. And yes, finally, the interpretation for antimicrobial activity is like the ANOVA table suggests that the model which we used is significant. And from this value, 54.88% of the independent variable can be estimated by knowing the concentrations and values of the independent variables, just say those chemicals and total phenol flavonoid content put together into equation can predict 54.8% of independent variable. So this suggests that the model which we used, where the relationship between the chemicals, individual chemicals and total phenol and flavonoid content and the antimicrobial activity is moderately strong. This is a regression equation. After putting those values into the equation, what we found was propolis activity quotient. So this number, what we is found is the median value and a combination of propolis from 3 different regions, multiple studies, hundreds and hundreds of studies coming together into the equation and then giving us a robust value. So when this can be referred as a median value for deriving your bioactivity quotient. Likewise, for antioxidant activity, we performed same study, just dependent variable was changed as an antioxidant activity in place of antimicrobial activity. And what we found was, again, here the R value was a bit lower than the antimicrobial study, but the model was still significant. And the ANOVA table suggests that the model is significant. and this is a coefficient and equation, correlation equation we derived. Um, so finally, to interpret the results, here again the ANOVA table suggests that the model is significant as the value is less than 0.05. The correlation here between the independent and independent variables is slightly lower, which is 22.38%. So what we can say is that our model suggests that the relation between the dependent and independent variable is moderately weak. So more studies, more robust data is required to increase the confidence and the predictability percentage in this case. So in this regression equation, what we found was the 49.53 as a propolis bioactivity quotient for antioxidant activity by using our model and the equation. So in the a review published in Journal of Apicultural Research, we proposed a classification system for propolis by using PBQ as a parameter to classify how your propolis is active against as a antioxidant or anti-microbial. So just for example, here this is that the PBQ, if it is less than 100, then you can class it as a class 1, and you can see here highly active propolis. And if the PBQ is more than 500, the number is more than 500, then you can say the propolis is highly possible that it will be less active against for its will have less antioxidant potential. So this we published, and then ultimately to conclude, what we found in our study was that the from the all climatic zones, the phenol content was higher than the flavonoid content, almost 1.5 times, and the correlation between phenolic content and the antimicrobial activity is better, and vice versa for flavonoids for antioxidant activity. So the independent variable in our model explains 54.88% of antimicrobial efficacy and 22.38% of antioxidant potential of the propolis. What we found was, as I mentioned in the earlier slide, galangin, cinnamic acid, and its derivatives are reported higher in concentration in tropical propolis. The apigenin content of tropical propolis was found highest among the propolis from other zones, and the higher the concentration of pinabaxine, pinacembrine, and chrysine was found in the propolis from temperate zone. So in the final conclusion, antibacterial activity quotient for propolis found to be 188.92, and for antioxidant activity, the PBQ was 49.53. So the one downside of this, as you can see, the chemicals specifically present in a particular location, just say Brazilian propolis has got typical potent molecules. So they are not included in this study. So that doesn't create a problem for this because we are looking at antioxidant and antibacterial activity. So, but if you're focusing on generating a PBQ for anti-cancer or leishmaniasis or any antiviral or any other activity, you need to identify which are the prominent molecules reported in propolis and see they are included in your study, because as an independent variable, that is very important so that you can have more confidence on the quotient which you derive by using the equation, but the concept can be same. Yeah, and if you have any question, please let me know, and then I will be happy to answer. So thank you very much for listening patiently. And I hope you enjoyed and be safe in this difficult time. Thank you.